All right, thank you everyone for joining to the second session of the Racing is the Clock and Valid DAO Journal Club. Today we are going to have an amazing scientist that my colleague Albert is going to introduce more formally, but he's going to be talking about exciting metabolic engineering and secretion of like molecules that are valuable for multiple industries. Hi, Stephen, and hi, Caroline. Thank you for joining us. So I will pass over to you, Albert, so you can introduce a little bit more about DAO and Rodrigo.
Yeah. Thank you, Ivan. So thank you all for joining. And of course, I mean, the intention of these journal clubs is to really highlight interesting researchers in the field, but also an opportunity for racers and community members of DAO to learn kind of like, okay, what are the most recent research results that are coming out from different climate biotech focused research departments. So Rodrigo here, he's actually one of the pioneering researchers that we've started to work with through DAO. And what we hope to accomplish is to get one of his projects funded, and also then help him into spinning it out into the university or to the industry, all through a community approach. So the community members are involved in helping out in the business development and the research due diligence, and all sorts of things. So Rodrigo, I mean, I think I could present you, but I think the right person to present is yourself. So why don't you kick us off with telling us a little bit about yourself, your work at Imperial, and the focus areas of your department.
Yes, sure. Thanks, first of all, Albert and DAO in general for the invitation. So I'm happy to be here and telling you a little bit about what I do in the lab, and especially about this particular paper. So I'm Rodrigo. I'm originally from Spain. I did a PhD in Spain in the area of metabolic engineering, working with the filamentous fungus and producing some lipid-derived compounds, mainly omega-3 fatty acids and some other nutraceuticals. Then I did a postdoc in Paris, working with Yarrowia Lipolytica, when I continued working in lipid metabolism and production of different compounds of interest, industrial interest. And 5 years ago, I started my research group at Imperial College London, where I'm based at the moment. So yeah, I did this small presentation just to introduce you to this particular paper that we're going to discuss. But I also introduced my group. So this is us. We are based in London, in South Kensington, so really central location next to the museums, Hyde Park, really nice area. And we are part of the Center for Synthetic Biology, one of the first centers for synthetic biology that was created in Europe. And it's quite large. There are around 40 different groups working in different aspects of synthetic biology, from mathematics to chemical engineers, bioengineers, people in life science, medicine. So a lot of different departments are involved in this multicultural or multidisciplinary environment. So just to set the context of what we're going to be talking about, and I'm pretty sure you all are familiar with these two things, but I'm going to give you just a brief definition of them. So synthetic biology and metabolic engineering, they are both trying to bring engineering concepts to the biology field, but they bring concepts from different engineering fields. For example, synthetic biology is bringing things from electrical and mechanical engineering, so trying to make biology more controllable, predictable, standardized as electric circuits that we have nowadays. While metabolic engineering is bringing concepts from chemical engineering. In chemical engineering, you have different chemical reactions happening in different tanks and some molecules converting into others. And that's basically what happened inside the cell with the metabolism. The metabolism is going to use enzymes to convert some chemicals into others. So in metabolic engineering, we tried these concepts and this mathematical background of the chemical engineering applied to cellular, the cell. So more generally, my lab is very interested in the transition towards the bioeconomy, so moving away from our current economy that is based on fossil fuels and non-renewable sources of energy, material, foods, and trying to accomplish all that using bio-based technologies, and more in particular, microorganisms. So microorganisms are very versatile. There are so many of them that it's very easy to find one producing the kind of compounds you want, and that compound can be a biofuel, a chemical, a material, a food, but the general problem is that they produce it in tiny amounts because they didn't evolve to produce what you want necessarily. So they don't produce enough to make a commercially viable process. So you really need to decrease the cost of production by synthetic biology. Try to maximize the production, the conversion of a substrate into a product, so the product can be produced in enough amount to reach market. And that can be done with synthetic biology. And one of the things we do in the lab is being very careful in the host we start with. Different microorganisms, they have different capacities given by evolution, and starting by the wrong organism can make your process and your design very complicated or even impossible, while choosing the right organism that is already good at producing what you want, or the precursors of the molecule you want, you will be more likely to succeed. And of course, there is a balance between ability to manipulate the organism, how much we know about it, and the natural capacities. And in this balance is what we need to play and decide which organism we work with. So we work with a lot of organisms in my lab. I'm going to focus today on Yarrowia Lipolytica, which I will introduce in a minute. We worked also in a lot of different compounds. Many of them are food related. So we work in aromas, flavors, colorants, antioxidants, lipids that can be used for biodiesel, but also as nutraceuticals. We work in protein production with various applications, vitamin production, materials. We also work in the use of different wastes as carbon sources, so we can decrease the cost of processes by making them using like cheap renewable resources like lignocellulosic materials or starch. So in my research group, we focus on 3 different areas. One is the development of new synthetic biology tools. Another one is the production of compounds of interest for industry. And the third one is the creation of synthetic microbial communities with application in biotechnology and biomedicine. So today I'm only going to focus in one example of the blue area here, so the bioproduction area. And the product we're going to be talking about are microbial lipids. So what's interesting about microbial lipids is that they can be produced from waste, as I just said. So you can take things like agricultural waste or urban waste or industrial waste and use this as a feedstock for the microorganism to produce the lipids. And those lipids, depending on the type of lipids, the amount of lipids, etc., can be used to make biodiesel, fuels, or nutraceuticals, or food, or feed for animals. You can make building blocks for polymers. You can make lubricants, pharmaceuticals, etc. So the amount of like industrially relevant product you can make from lipids is enormous. And you might be wondering why microbial lipids, if we can get lipids from animals or plant sources. So another reason is microbes don't compete with food today. So if you use animal or plants, then you are removing resources from the food chain to make something else, which is not ideal. Also, microorganisms, they have short process cycles, which means you can duplicate the biomass of a microbe in 20 minutes if it's a bacteria to 1 hour and a half if it's a yeast. While plants, they take cycles of months to create a new plant, etc. They are independent of seasonal and climate. We provide the right condition in the bioreactor, so it doesn't matter if it's snowing or it's in the middle of the desert. And they can be scaled up just by building new bioreactors. So we don't depend on the land availability or water resources. It's, as I say, we can use cheap carbon substrates, and they are also susceptible to metabolic engineering. So we can really manipulate them to produce specifically the lipid that we want. Okay, and then I'm introducing a little bit the organism this work is about, which is Yarrowia Lipolytica. So Yarrowia Lipolytica. So when I started to work with it after my PhD or during my PhD, it was very niche research. There was only like 4 or 5 research groups in the world doing like tools or processes with this organism. Now it's becoming more and more important, and we know more and more its metabolism, so we are able to engineer it. It naturally produces relatively amount of lipids, so you can see here the cells and the lipids stained in green. So it's been considered like a good lipid producer, and because it's a good lipid producer, it can be a good biotechnology host to make fuels and chemicals coming from these lipids. So as I was saying, it's becoming more and more popular. This is a graph that's showing you the trend of use or publications of this organism. It's exponentially growing. There are more and more tools that are developed, more and more academic labs that are working on this, and more companies that are basing their processes on this interesting organism. So just to set the context, when we think about the bioprocess, we go from carbon substrate or a feedstock that is used for the fermentation. So this feedstock is converted into products, and then the products need to be separated. The downstream part of the process is often the most expensive part, and it can be up to 70% of the total cost of the bioprocess. So if we can do engineer to favor the recovery, isolation, purification of these products, we can really save a lot of money in downstream processes. And that's exactly what we aimed in the paper that we're going to talk about. That is the one that you were circulated before, and you may have had the opportunity to read or to look at the figures. If not, I'm going to go through it quickly now before you have time to ask any question. So what we wanted to do is to not only to produce the lipids that I show here that Yarrowia is able to produce, but also help in the downstream process by secreting this lipid outside. So because these lipids are inside the cell, if you want to recover them, you need to break the cells down. And for this, you need energy, either mechanical energy or enzymatic activities that in both cases contribute to the economy of the process or make it more expensive. So if we can really secrete the lipids outside, we can make lipid production more affordable. So the problem is that in nature, lipids tend to stay inside. And so this is how lipid metabolism works in Yarrowia. So the carbon source, in this case glucose, is incorporated into the cell, and through different chemical reactions, there's going to be converted into these lipids. So the fatty acid or the acyl-CoA are going to join a G3P to generate 3-acylglycerides, which are the main storage lipid form in the cell. So these lipids are neutral lipids, so they cannot cross membranes. They are not going to be released at any point. But the cell has mechanism to reuse these lipids. When the cell is hungry again, it needs to reuse this storage of energy. And for that, they use these intracellular lipases that release the fatty acids. And these fatty acids are usually quickly reincorporated into acyl-CoA or degraded in the beta oxidation to generate energy. But these free fatty acids are amphipathic, which means they can cross membranes, contrary to the 3-acylglycerides. So what we decided to do is to block these two processes to favor the secretion of the free fatty acid across the membrane. So by doing that, we generated a strain that was either blocked in the first step or both steps. And you can see here the amount of fatty acid that were produced were significantly higher. And you can start seeing in the microscope these lipids floating around, and this turbidity that comes from the emulsion of lipids in the liquid phase. So we wanted to produce even more lipids now that we knew we were able to secrete the fatty acid. So we did 2 different strategies to achieve that. The first strategy, we tried to maximize the natural process by overexpressing some DGA genes. These are genes that are going to do the last step in the production of 3-acylglycerides, and we know that if we overexpress this gene, we produce more storage lipids. And overexpressing as well the intracellular lipases, TGLs, we theoretically, we will convert more glucose into more lipids into more free fatty acid, and this free fatty acid would potentially go out. And this is what we observed. So here you have the different genetic modification we did, and you can see how this is the strain I showed before that produced some fatty acid outside the cell. And then when we express the DGA2, it starts to increase the intracellular lipid content. So this part is intracellular, the top part is extracellular. So the DGA2 really increased the intracellular, but not the extracellular as we expected. But when we start to express the intracellular lipases here in different combinations, you can see how the intracellular lipids are being degraded, and proportionally the extracellular lipids are being increased. And this reached 3 grams per liter of lipids outside the cell. And you can see very easily, and you can start imagining how this is going to favor the downstream process, because the lipids simply accumulate outside, they clump together, and they can be very easily recovered just collecting them from the surface. This is how they look like in the microscope. These are the non-engineered strains with their lipid bodies inside the cell. And this is the engineered strain where you can start seeing these green dots. That means the lipids are being secreted. So then we tried a different strategy. This strategy, we were trying to mimic what happened in bacterial systems. In bacteria, there are no intracellular lipids. So we've removed all the genes that make triacylglycerides in Yarrowia. So what is going to happen if we do that, if we accumulate acyl-CoA, and the acyl-CoA is known to regulate the pathway in a negative way. So in order to remove that negative regulation, we had to express some enzymes called thiosterases that are going to convert the acyl-CoA into free fatty acids. And that free fatty acid is the product we want because it's the one that can be secreted. So again, we generated cellular strain by genetic engineering. And here you can see, if we focus on the part here on the left, you can see how when we remove, this is the Q4 strain, when we remove these 4 genes here in red on top, you can see how the intracellular lipids decrease, cellulars as well. And this is because of the feedback regulation I mentioned. And when we start expressing these thiosterases from different origins, we start to see an increase in the extracellular production. This is because the free fatty acids are being released by the thiosterase. This part on the right is not very relevant. It's just telling you that these thiosterases, they don't help secreting unless these 4 genes are being removed. So this is how the cells look like in the microscope. The control strain looked the same as before, but the engineered strain has the lipid outside. Differently to the one I just shown, in this example, there is no lipids inside the cell. If we go back, we can see that in this case, there were lipids inside and lipids outside. But in this case, almost even 85% of the total lipids are being secreted. So just the final part of the paper is explaining how we can try to scale that up and to move to a bioreactor. So all the previous experiments were done in flask. And when we go to bioreactor, we can see how we can really increase the production titers to 15 grams per liter. We can see much more lipids being secreted, these big green droplets here. And we also performed some two-phase bioreactor where we added a dodecane phase. This is an organic layer that allows the extraction of the lipids, and by doing that, we managed to produce even more gram per liters of total lipids, around 20 grams per liters. But something that was interesting about this is that for the first time, this allows us to produce more lipids than biomass. Before secretion, you were constrained by the cellular capacity to the amount of lipids you could produce. Now, because we can secrete the lipids, we can produce more lipids than biomass. And this is what this graph is showing here, that if you account the number of lipids that you produce per number of biomass, you get a number that is higher than 120%. So with this, I would like to acknowledge my current lab members, but also the lab members of the lab where I did this project, this paper we are talking about during my postdoc, also my current funders. And I would like to thank you, all of you, for your attention, and I will be very happy to answer any question.
Yeah, cool. So now we're going to move to the discussion and Q&A with Rodrigo. I think that as Rodrigo has a long track experience and such an amazing technical domain on metabolic engineering and synthetic biology in general, I think it's the right opportunity to make questions about the paper, and also if you have questions in general related to his research, please go on. Sorry. Thank you for raising your hand, Andres. Please go ahead to make your question to Rodrigo.
Thanks. Thank you so much, Rodrigo. Reading your paper, I noticed there's a researcher called Xochitl News from Guadalajara. I just wanted to know how if she collaborated or how she participated in this research.
So that Xochitl was like a visiting researcher. She was doing her PhD in Guadalajara, and she came to the lab where I was in Paris to spend, I think, something like maybe 4 or 5 months. And so we were working in another project with her, but she helped me in this project in the bioreactor part of it, and that's why she's a co-author in this paper. We published another paper where we were using actually lignocellulosic materials as a carbon source for Yarrowia, and that's the expertise of the lab in Guadalajara. So we were bringing samples from agave, from the plant that is used to make tequila. I don't know if, so you probably don't know, but for every liter of tequila that you make, there are 10 kilograms of waste biomass that has no application, it's just disposed in the field. So we wanted to valorize that biomass to make a product, in this case, a biodiesel. So that was the work we did together with this Mexican lab.
Okay, thank you.
So exciting. By the way, Andres is from Mexico, so I can notice why he was curious about that. Someone else, please raise your hand. If not, I have some questions, but I will leave mine to the end. Is there any challenge with the downstream of this lipid secretion? Like it perturbs like the bio reactor or the biomass after you start to have secreted like an amount of lipid?
That's a very good question. So we tried 2 fermentations. So the first one was just growing the cells with liquid media, and in that case, you can see the lipids floating. Sometimes they get a little bit attached to the walls, but you can easily recover them when you finish your fermentation. The second type of fermentation we try is the 2-phase fermentation, where we add a dodecane and anacane phase that is insoluble with water but solubilize the lipids, and that's supposed to be an easy way to recover the lipids while they are being produced. And in that case, which is the one in which we produce more lipids, we found a problem, and it's the Yarrowia like hydrophobic substrates. So even though in this case Yarrowia was not really eating this dodecane phase because there was enough glucose in the media, Yarrowia was still stuck to this hydrophobic surface because of the surface of Yarrowia is also hydrophobic. So at some point when you finish the fermentation, you have not a 2 layers fermentation, but a 3 layers fermentation, where you have the hydrophobic phase, the Yarrowia phase, and then the liquid phase. So the separation was not as good as we would have liked to, but there are room for improvement there, like changing the hydrophobicity of Yarrowia, etc.
Okay, cool. And we have Bethan Hiley asking, is it possible to combine the 2 strategies?
So by the 2 strategies, do you mean the 2 strategies I described in the paper, the 2 metabolic engineering strategies, or the 2 fermentation strategies?
The 2 strategies, so the one that is upregulating DAG, and the other one where you've added the THIOs from the bacteria.
Yeah, so let me maybe go back and share my screen. So this strategy, the problem and the reason why I don't think it can really be combined is because of this part that I kind of skip. So I understand your question, it's a very good question, but this part here on the right side of the graph demonstrates that probably it won't be compatible. And what we are seeing here is that if we don't have the Q4 strain, which means these 4 deletions, then we don't really see secretion. We see not increase with the thiosterases either. So the thiosterases don't really have any effect if you compare this first one here with the rest. So the other strategy really depends on the formation of the TAGs. So the way of combining will be to overexpress these 2 genes and also overexpress the thiosterases, but this suggests that it's not going to work.
Yes, thank you.
So that's something interesting about that actually was part of the initial idea of the project, is that thiosterases are also very interesting enzymes because they have preference for some fatty acid lengths. So you can express thiosterases from different organisms, as we did, and some of them are known to release specifically short-chain fatty acid or medium-chain fatty acid. So we were trying to see if expressing different thiosterases we could see different length, and we tried this also in this scenario, but it didn't really work like this. And then we found out that that's mainly related to the differences between the different systems where these thiosterases have the preference for the chain length are either bacteria or plant. So they work in mitochondria or bacteria, and in yeast system, these thiosterases cannot really access the fatty acid synthase complex as well.
Okay, thank you so much, Rodrigo. It's very insightful. Yeah, and I think here in the audience, like, I'm not sure if someone is studying precisely more about the chemistry or metabolic engineering or bio, but it's good to see like the holistic approach of how to, integrate.
I have a question regards to if there is a theoretical limitation to let's say the system and the continuous bioprocess. Is this something that you look into? Like, for example, how long can you sustain the continuous bioprocess before you just have to stop at harvest and basically repeat it?
So I guess there's a big difference between the theory and the reality. The reality we don't know. We haven't really investigated that yet. In theory, there is which is called the maximum theoretical yield, that is telling how much you can produce if all the carbon you're feeding into the system is translated into fatty acids. So theoretically, by growing your cells first and then using them in a continuous way, you could potentially achieve these maximum theoretical yields, because you don't need to use the carbon source to build more new biomass every time. But that's one of the advantages of these kind of processes, is you don't waste energy and carbon into making new biomass. You can just do that once and then continuously produce the lipids. In theory, but we haven't explored for how long you can do that, or if you can immobilize the enzyme, the cells, and just flow liquids through them over time. There's a lot of possibilities, but they haven't been investigated.
Okay, super quick. I guess I'm just really curious about your opinion. How long do you think it will take before this research actually is part of a real industrial process?
That's a good question. Something that I think COVID has demonstrated to humanity is, like, if you put enough resources into a problem, you get a solution based on science, and I think that will be applied to almost any problem we want to solve with biology, right? If we throw enough money on it and there's enough interest from society and politicians, then it's achievable, and that, I believe, it applies to any applications with biology. In this case, I guess it depends on the specific lipids you produce in the end. So I think the system can be improved in terms of yields. The production are not bad, are on the scale of 20 grams per liters, and around that is where you can consider that's okay for scalability. But still, lipids are very cheap when you compare with like plant-based lipids, and either you increase the value of the lipids because they have a functionality that can be used for infant formula, for milk substitutes, or else nutraceuticals like omega-3 fatty acids, etc. If you just want to do bulk lipids for biodiesel, I think we cannot probably compete with plant biomass at this stage, just because the infrastructure requires a lot of investment, while the infrastructure for biodiesel from plant is already there. I guess after there's enough investment in the process itself and bioprocesses in general, everything's going to be cheaper to implement. So that's a long answer for a short question, but I would say putting enough resources in this, in a couple of years, you can increase the production. You can see if there is enough levels of product to explore commercialization, but always with specific application in mind.
Yeah, that's the question that I'm wondering. Like, do you have any application that you see more potential than others? Like, at this point, I saw that you have from biofuels to food, cosmetics.
I mean, we originally started working in biofuels for this project because at that point it was a big thing. There was investment in that. Even the airplane companies, they were investing in this. But then the prices dropped, and then we tried to find alternative applications. Anything related to biomedicine, always going to have high value. Now nutrition is getting a lot of traction, so applications in nutrition, I think, are going to be feasible in the next few years. Fuels are getting more expensive now with the current situation, at least in Europe. What is going to happen next, we don't know, but maybe there's a room for microbial biofuels as well.
Cool. I can see that Yang is raising his hand. Please unmute and make your question, Yang. Thank you.
Hi, Rodrigo. Just one more general question is that I'm not from a metabolic engineering background, more like a simple part. So I'm curious that what are the challenges when you are developing tools in the Lipolytica?
So Lipolytica is I wouldn't say it's a difficult organism to work with. I would say the opposite, but it's not straightforward either. So I've seen many people trying to work with it without asking around how to work with it and struggle. But once you know how to work with it and you ask the right people for tools and protocols, then it's a nice organism to work with. Of course, there are challenges like homologous recombination is not very efficient. So you need to be a bit patient about finding the right mutants. Maybe you need to screen a bit more clones that with Saccharomyces cerevisiae, for example. You have more gene duplication, you have a bigger genome, and that can be complicated sometimes. Because the genome is bigger, we don't know the role of some of the genes in Yarrowia, so we still need to develop a little bit more of fundamental knowledge about what all these genes are doing. So yeah, there are challenges like in every other organism, but there's still advantages in many other things.
Yeah, thank you.
I think it's expanding on that. I think it's a good example of how synthetic biology and the synthetic biology community can domesticate an organism that 10, 15 years ago it was very niche. There was no tools, there was no knowledge, and now there are a lot of information out there. From synthetic biology, synthetic biology tools, CRISPR technologies, etc. So demonstrating once more that if we put efforts, we can achieve almost whatever we want.
Yeah, I think there is a huge potential of using non-model organism and untap a lot of solutions that for the problems that we face in the lab. But also like biology. Like I personally think that we are understanding biology, but still we're scratching the surface, and starting with the non-model organisms, it requires to be much more patient, as you said, and the resources. I'm not sure, do you have any struggle to canalize resources because of using a non-model organism?
So what was the question we struggle with?
With canalize resources or grants or like funding, because that reason, or it's you will say that like scientific community is open to explore.
I think with Yarrowia is now more open. I think a few years ago it was more difficult because people didn't. And I still have some grants rejected because people say, "But why do you use Yarrowia?" Which I still don't understand. When you can just look at the papers and you can see you produce 10 times more of the same compounds in half the time. But yeah, some people are still like hesitant to explore new organisms because it's true, it's challenging, and there are other problems you can face. But I think also the opportunities that it opens compensate. So I would say sometimes it helps, sometimes it doesn't, depending on the reviewers that you get.
Yeah, I heard you talk about like lignin cellulose at some point. I was wondering if you were using that as a substrate.
So I think lignin is the only part of the hydrolysate we haven't tried. So we tried the pentoses, the silane, cellulose is the glucose, fructose, etc. We never tried lignin as carbon source. We know so many of these lignins can be toxic to cells. Probably in high amounts it will be toxic for Yarrowia as well. Well, Yarrowia is quite good at tolerating toxicity, so it's something that's probably worth exploring. Lignins are used as carbon source, has been used in different organisms with different levels of success, and in my opinion, they are very interested as precursors of some specific molecules that contain some rings in structure that could like skip some steps in the metabolic pathway if they use some lignin precursors to build them. So probably as a single substrate is difficult because of the toxicity, but maybe as a co-substrate it will be interesting for specific type of molecules.
Cool, thanks. It's a great paper.
We have a comment from Stephen that he unfortunately needs to leave now, but that he appreciates a lot how you are domesticating microbes, sorry. And he mentioned that solid state fungi probably are best for using lining. Rodrigo, thank you so much. Albert, thank you so much for organizing this journal club. If there is something else, Albert, that you wish to share before we close.
Yeah, so thank you all for joining. It's I mean, I think there's an underestimation of how much value these sessions bring to the researchers, and also if you know of any like future researchers that you could nominate or recommend to the journal club, I mean, even can try to get hold of them and bring them here. And also I think also Rodrigo's group are in the constant lookout for PhDs and postdocs. So feel free to reach out to Rodrigo. Rodrigo, if you want to maybe drop your email in the comment section.
Yeah, do that. I'm actually looking for a postdoc from tomorrow, so if you know someone to join.
We can circle that on the Slack channel where we have like the full.
You can write a little blurb, Rodrigo, and send it to me and Ivan, and we can post it on both ValiDAO and Race Against the Clock Slack.
Yeah, cool.
Everyone for the question, all of them were great.
Yeah, and just before we close, let's remind that the bioeconomy is not going to be built alone. Like, it's going to be people like you and me. So like participating in the journal club and our initiatives that we are pushing are intended for helping our growth as biocontributors. So I hope that your dreams with biology are very exciting and that you find the means through our community to make them real. So
Yeah.
thank you so much, everyone.
If I can just drop one final thing, really is, I mean, if you're personally interested in supporting the cause of ValiDAO and believe biomanufacturing should be democratized and easy accessible, you're welcome to join the community. The community is filled with researchers like you, serial bio-entrepreneurs, science communicators, really all working together to progress democratic funding and access of climate-focused biomanufacturing research. Franco, you recently started helping out in the deal flow group. So if you want to, maybe put a comment in or anything like that of how your experience have been, that would be super useful.
Yeah, I think it's been really great. Like I've been learning a lot from the people that are especially in Discord, because there's a lot of ideas bouncing back and forth, a lot of ideas, not only from the academic side, but from the business side, also how to communicate. I feel that it's really valuable. And we met with some people to evaluate it, if a project is good or not for continuing. So I think it's great to join the community and be part of it.
You need to set me up, Albert, because I already did download Discord, but that's a whole new world for me. I know that it's a kind of Slack, but
yeah, it's a little bit of a barrier to entry getting familiar with Discord. But once you get used to it, it's really streamlined and efficient. But yeah, if you need help with getting started, you can always reach out.
I'm sure that it's not hard. It's just me being like busy and lazy to just go and figure out things. But now that I'm going to see you, let's make it an objective. I'm going to get set up on Discord. All right, thank you, everyone. Thank you for staying a little bit late, 5 minutes. Stay safe and let's continue building the bioeconomy. Thank you, Rodrigo, and thank you, Albert. Thank you, everyone. See you later.
Thank you.
Well,
Bye.
Bye.